Hello everybody, I am Dr. D.S. Thaure. Now I share the screen of today's lecture in front of you. Please wait. Okay, welcome to the 8th lecture of course number Path 365. Title of this course, Diseases of Field and Horticultural Crops and Their Management Second. Okay, so in today's lecture, we will discuss with you about diseases of wheat. In previous lecture, we already discussed about rust disease seen in wheat. Okay, so in today's lecture, remaining disease we will discuss with you. Okay, so first of all, loose smut of wheat. Okay, so this loose smut of wheat disease is the most prominent disease seen in wheat crop. Especially this disease generally seen are carried through infected seeds sown in the fields. Okay, means the fungus is carried over in the seed as a dormant mycelium. When such infected seeds sown in the fields, so after a few days, these infected seed germinate, so these mycelium become active within present in the seeds. So it mycelium grows along the plants, and when the panicle is produced, the mycelium. Okay, so this mycelium reach to this ovaries. Means first initial infection generally seen at the time of the flowering period of the wheat crops. Okay, means these infected seeds become germinate and grows, means this mycelium grows along with the plants, and when the panicle is produced, so this mycelium attack to this developing ovaries. Okay, so this developing ovaries transformed into the mass of spores. Okay, means these infected airheads on diseased plants emerge out from the flag leaf earlier than the earlier than the healthy one plants. Means this infected loose mud airhead is easily located uh, from these fields, because this infected airhead is produced from this flag leaf of this plant is earlier than the infected plants. Means this infected airhead is partially or completely transformed into the smut sori. Okay, so this smut sori generally covered with the thin silvery membrane. So this thin silvery membrane structure become burst from these infected airheads generally produced from the flag leaf of the plants. Okay, means in this case, this individual ovaries is totally transformed into smut sori, or the maximum number of the ovaries generally converted into the smut sori. Means the some parts of the airhead transformed into smut sori, or whole the portion of the airhead containing the ovaries is transformed into smut sori. Okay, means in this case all the reproductive organs get converted into smut sori except ons portions. Means at the maturity stage of the fungus, the infected smut spores release the somewhat brown to black spore mass is easily released or blown out from these infected airheads. And this blackish or brownish powdery mass just seen on this lip portion or seen on this infected airhead portions. Okay, means this individual airhead transformed partially or completely into smudge sori. So this smudge sori having the brownish to blackish spore mass are produced, and this spore mass is released through this infected ovaries and just fallen on the healthy one airhead of the wheat crops. So in this way, transmit or spread the disease from infected plant to the healthy one plants. Okay, so this is the symptoms of the loose smut disease seen in the wheat crop. Okay, then causal organism, that is a Ustilago tritici. Okay, so this Ustilago tritici fungus produces the spores. Just to see this disease cycle of the Ustilago tritici. Okay, means these spores generally olive brown in nature and spherical in shape. Okay, so these spores become germinate and to produce the germ tubes, and these germ tubes generally infect to the healthy one ovaries. Okay, means about 3 weeks old plants, the hypa or mycelia reach to the lower end of the hyphae and pass around the bottom of the endospores having the seeds, means finally reach to the embryos. Okay, so this mycelium invades parts of the embryo in the seeds. Okay, so this mycelia become dormant in these infected seeds for long durations, and these seeds generally sown in the fields. So 100% this dormant mycelium become active, and this mycelia just grows or becomes systemic in the growing parts of the wheat crops. Means this mycelia become persist in the growing wheat crop at the means reach up to the development of the ovaries in the airheads. Means at the time of the development of the ovaries, this mycelia become active and just insert to this developing ovaries. So this developing ovaries is totally convert into smut sori. Means these spores generally produce the smut sori, and this smut sori having the spores generally black or brown in nature. Means this airhead is totally converts into smut sori having the blackish or brownish spores. Okay, so this suppose this blackish or brownish mass is blown with the help of the wind, so just remain rachis portion or this only airhead is remain without the formation of the grains. Okay, means this infected plant parts or infected airhead debris in the soil, or this spore mass just fallen in the soil, so these spores germinate and to produce the four-celled promycelia, means the formation of the teleospores. Okay, so in this way the sporidia acts as a secondary source of infection, and primary infection, that is these infected seeds or these infected plant parts debris in the soil, means particularly infected airhead having the spores deposit or debris in the soil, so 100% disease is produced. Means in this case primary infection, that is internally and externally seed-borne fungus and become systemic, and secondary infection, that is this sporidia disseminated through wind or air currents, and in this way spread the disease infected plant to the healthy one plants. Okay, so this is the primary infection, secondary infection, and etiological parts of the Mustilago triticale. Okay, then control measure for this loose mud disease. So first, the use the healthy one seeds or disease-free seeds for planting purpose, because primary infection takes place through the infected seeds sown in the fields. So before the actual sowing of the seeds, so these seeds generally treated with the hot water treatments, means take the seed and just dip it in the cold water for 4 to 6 hours. So these dormant mycelia actually present inside the embryo portion of the seeds. So these dormant mycelia become active due to the cold water or due to the favorable conditions. So this means after 4 to 6 hours, uh, remove the seeds from the cold water and again dip it in the hot water having the temperature 52 degrees Celsius for 10 minutes. Okay, then after 10 minutes, these seeds is removed from these pans, and these seeds is again treated with the carboxyl, that is Vitavax, at the rate of 2 gram per kg of seed, or carboxyl plus carbendazim at the rate of 2.5 gram per kg of seed. Means in short, firstly these seeds are treated with cold water, then again treated or dipped in the hot water, and finally these seeds are treated with the, some fungicides. So eradicate or to eliminate the mycelia present inside the embryo of the seeds. Then solar heat treatments. Okay, so in this case they soak the seeds in cold water for 4 hours and then immerse in the seeds in hot water. Sorry, these seeds uh, remove from the pan from of the wheat seeds, and these seeds generally spread in a thin layer of on the threshing floor or any galvanized iron seed. Means just take the galvanized iron seed and these uh, deep seeds just spread over the iron seed or any galvanized seeds during the summer month, or in especially 12 to 4 o'clock p.m. Means during this period maximum sunlight is reached to these seeds. So that's why these dormant mycelia just killed due to the sun rays. Okay, so in this way you can manage the loose mud disease. And last one, growing resistant varieties, that is kalyan sona or other variety is grown by the farmers to avoid such a infection seen in the wheat crops. Okay, so this is the loose mud disease seen in the wheat crop. Then canal bunt of wheat. So this is also severe disease seen in only few location in particularly Himachal Pradesh, Uttar Pradesh states in India. Okay, so in this case, look at these pictures of the infected airheads having the grains are converted or individual grains are infected. Means these pathogens attack to these airheads at the time of the grain developments. Means at the time of the grain development, the fungus directly attack to these developing grains. Okay, so these developing grains is partially or completely converted into black powdery mass. So in this case, only few portion or few surface of the seeds is converted into black powdery mass. But in this case, near about 70 to 80% of the seed is transformed into smart powdery mass. Okay, means the infected airheads in which only few grains is converted into smart powdery mass. no totally airhead is converted into smut powdery mass. But in case of the loose smut, partially or completely airhead is converted into smut spores. But in kernel burnt, this only few grains is converted into smut sora, and this grain is partially or completely converted into smut sora. Okay, means such partially affected seeds may or may not be germinate, but completely converted seeds is do not germinate and produce the plants. Okay, means the spore mass remain covered in the pericarp, but after few days or after latter this stage rupture the seeds and releasing the black powdery mass, and this black powdery mass deposit on the another healthy one grains in the same airhead or other nearby airheads, and this airhead having the seeds is converted into blackish spore mass. Okay, means here this disease is also known as the kernel burnt. Okay, means first of all this infection generally seen in the Karnal district of the Haryana states. So that's why this burnt disease is also known as the Karnal burnt disease. Okay, means this individual grain convert into blackish spore mass. So this is the symptoms generally develop in the individual grain in the infected area of the wheat crop. Okay, then etiology of the Neovacia indica. So this Neovacia indica uh, fungus is responsible to produce the kernel burn disease in the wheat crops. Okay, so just to see this disease cycle means these teleospores. Okay, so uh, these teleospores in soil become germinate producing the means suppose this infected airheads having the few grains are converted into blackish spore mass, so these bunted balls or bunted spore mass or the infected grains just debris in the soil or just these seeds is used for sowing purpose, so 100% this is produced means these seeds become germinate, so this mycelia become active or persist within these growing parts of these wheat crops, so finally these germinating seeds and germinating seedlings pursues the mycelia, and this pursues the mycelia become active and directly attack at the time of the grain development stage, so this individual grains convert into galls or just burnt portions having the blackish spore mass. Okay, so in this way primary and secondary infection takes place, means secondary infection, that is this black spore mass having the sporidia. So this sporidia generally disseminated from these infected airheads and deposit or fallen on this healthy one airhead. So in this way, this healthy one airhead become infected. And primary infection, suppose this infected seeds used for sowing purpose, so 100% disease is produced. Means these infected seeds or any plant parts debris in the soil, so this teleospore become active or become germinate under the favorable conditions and to produce the haploid sporidia. So this H-shaped haploid sporidia generally fused each other and directly infect to these healthy one grains. So this is the disease cycle of the Neovacia indica. Okay, so this is the same disease cycle of the Neovacia indica. Means these infected seeds deposit in the soil. So these infected seeds having the spores become germinate under the favorable conditions and to produce the H-shaped sporidia. And this H-shaped sporidia directly fallen on the healthy one airhead of the wheat crop. So this healthy one airhead become infected. So in this way, spread the disease through this sporidia by means of the wind or air currents, or this secondary sporidia directly infect at the time of the flowering. So these flowers do not develop the seeds. Okay. So in this way, this primary and secondary infection takes place through this fungus. Okay. Then control measures, that is a growing resistant varieties. So this is the number of the resist variety grown by the everyone farmers against or to avoid such infection seen in the wheat crop. Then follow strict quarantine measures. Means during the export and import of wheat seeds from other countries or state to states within the India. So firstly check the these wheat seeds whether it is infected or not. Okay, suppose these infected seeds, so avoid these seeds or reject the seed lots from these import or export materials. Then use the disease-free seeds for sowing purpose. Then judicious management or application of the nitrogen fertilizers. Then adjust the date of sowing to avoid the infection to wheat crop at the time of the flowering or grain development stage. Then intercropping with the gram or lentil crops, then seed treatment with the carboxin or Vitabax or Thyram at the rate of 3 gram per kg of seed, means to kill the mycelia present on or inside the seed by using the fungicides. Then spray the crop with carbendazim at the rate of 0.1% or carboxin 0.2% or mancozeb at the rate of 0.25% or bitter tanol at or before grain development stage of the fungus. Means this fungicide generally applied before or just after the grain development in the particularly airhead of the weeds. Means this protect the airhead from this neovacia fungus by applying the some fungicides. Okay. Then third one most important disease seen in the wheat crop, and this is the most common disease seen in the every one variety of the wheat crops. Okay, so this leaf blight infection generally seen on the lowermost leaves of the wheat crops. Means first initial infection of the fungus seen at the basal side of the leaf. Okay, so on the surface of the leaf, the small oval discolored lesions seen on the lower side of the leaf. Means this infection generally seen uh, where the plant generally 7 to 8 week old, means this infection generally seen at the seedling stage of the wheat as well as the crop is mature. So initially infection seen on the basal side of the leaf having this small oval brown to gray spots seen on this leaf. So under favorable conditions, these small brown to somewhat gray spots just spread or enlarges over this infected leaf. So this leaf having the maximum portion of the lesions or maximum portion of the leaf covered with this brownish lesion. So in this way, this leaf is totally infected, and this leaf is become necrotic in appearance. Means look like as a burning appearance, or sometime this infected leaf or burning leaf generally seen from tip of the leaf to the downward directions. Means this drying of the leaf is seen from the tip of the leaf to the downward directions. Okay, so these lesions varies in the size, means near about 1 centimeter or 2 centimeter, depends upon the size of the leaf and inoculum load of the fungus. Okay, means most favorable conditions, these lesions coalesce to each other and cover the maximum area of the leaf, and these lesions is known as the blight lesions. Okay, so this infected leaf is totally defoliated from this uh, plants. Okay, so same, that is a small oval brownish to gray spots also be seen on the leaf sheath, stem, as well as the formation of the airhead or surface of the awns and glooms of the airheads. Okay, means the spikes are infected at the time of the pre-anthesis developments or pre-anthesis stage. So these spikes is infected in the early stage, so that's why no any seeds is formed within these airheads, or these seeds may be formed so at the time of the glooms, or means on the surface of the glooms, brownish spots are seen or brownish lesions are seen. So these glooms having the seed is also infect and showing the brown spots are seen. Okay, means these brownish lesions or discolored seeds is also seen within this infected airhead. So in this way, maximum grains infect by the fungus and showing the brownish or discolored seeds is produced from this infected airheads. Okay, so in this way, the infection generally seen or spread from leaf to the leaf seed to the stem portion, and finally reach to these airheads having the grains. Okay, so in this way retard the quality and yield of this wheat crop due to the small spots seen on the grain portion. So that's why these grains having the less market value. Okay, so this infection generally seen due to the fungi causing the Alternaria triticina. Okay, so this Alternaria triticina develop the spores and spore bearing hyphae, means these conidia. So this is this asexual spores, that is a conidia, generally formed at the tip of the conidiophore. So you see this individual conidia. So this conidia generally formed in the acrogenous in natures and born singly or maybe chain at the tip of the conidiophores. So this conidia generally tapered at the one ends, means one end of the conidia just tapered, and other end of this conidia just rounded, means one end of this conidia rounded and other end just beak like structures. Okay, so these spores or this conidia generally pale brown to dark brown in natures, and this conidia having the two types of the septa is present, means transverse septa and longitudinal septa. Means this is the transverse septa, 1, 2, 3, 4, 5, 6, 7, 8. Okay, means near about 1 to 10 transverse septa is present within this single conidia, and longitudinal septa means 1 and 2. Longitudinal means vertical septa is formed within this conidia, and this longitudinal septa is near about 0 to 5 in number. Okay, so these two type of the septa is present within this brown color conidia. Okay, means in this case primary infection takes place through the infected seeds or infected plant part debris in the soil, and secondary infection, that is conidia, spread through wind or air currents. Okay, then how to control this disease may be seen in the wheat fields. So in this case, collect and destroy the affected plant parts and burn it properly to avoid the primary source of infection. Then soak the seed in water for 4 hours followed by deep in hot water having the temperature 52 degrees Celsius for 10 minutes. After the 10 minutes, remove such a seeds from this pan and kept in the said conditions. And then this seeds is treated with some fungicide, that is Vitavax, at the rate of 2 to 4 gram per kg of seed. And these seeds is used for sowing purpose. Then growing resistant variety CO25, Sonalika, or Nantika, then E6160 and K7340, or these brownish spots may be seen on the leaf or airhead portion, means on the surface of the glume. So immediately spray the wheat crop with foliar fungicides, that is Zineb or Mancozeb at the rate of 0.2%. So you can reduce the disease incidence on the wheat crop by applying this fungicide uh, at the uh, 2 or 3 sprays should be followed or over these crops or during these wheat cycles. Okay, then last disease of the today's lecture, that is air cockle of the wheat. So this air cockle disease generally seen in the wheat crop due to the nematodes, not due to the fungi or bacteria. So this infection generally seen due to the nematodes. Okay, so nematode is a pathogens infect the crops and produce the symptoms. Okay, means these nematodes having the larva directly infect to the wheat seedlings as well as the mature plants and develop the typical symptoms on the seedlings as well as the mature plants. So initially symptom, that is the drying of the plants, then uh, wrinkled and twisted the leaf is seen on the younger plant or on the seedling stage of the plant. Okay, then retard the height of such infected seedlings as well as the mature plants. Then reduced and irregular head or airheads, and this means this infection generally seen on the mature plant. So this infected mature plant develop the airhead, but this airhead is also infect, and this airhead having the small in size, or this airhead is wrinkled, deformed. So such type of the infection seen on the airhead portions. So this airhead is looks like as just cockles. Okay, so cockles, okay, means these light to dark brown round balls generally seen on the infected airhead as compared to the healthy one grains. Okay, means this infection generally seen on the seedlings, means on the leaf portion as well as the mature plant develop the airheads. So this airhead having the seeds is also converts or infected. So these infected grains develop the round galls, and these round galls generally dark brown in colors. Okay, means finally growth of this plant is also reduced or stunted the growth of these plants, means this airhead of from these diseased plants do not emerge properly. Means suppose this infected plants develop the airheads, but this airhead is not properly developed, or this awns and some other portion of the airhead is totally infect and may be twisted in appearance. Means this twisted appearance on and airhead portion is seen. Okay, so in this way much portion of the airhead is twisted and converts. Okay, so this type of the infection, and this airhead or this plant easily located from the wheat field, because this all the leaf and airhead means whole plants is infect and twisted in appearance, and this twisted plant or curling leaf of the plants is easily located from these wheat fields. Okay, suppose these infected airheads develop the grains. So this grain is totally converts and to form the brownish galls are produced. So these galls having this some juvenile substance secreted by these larvae of the nematodes. So that's why this juvenile substance is also seen or produced inside these infected seeds or infected galls produced within this infected airheads. Okay, then causal organism, that is Angunia critici. So this nematode is responsible to produce such infection in particularly wheat crops. Okay, means this Angunia critici, this male and female nematodes is distinguishable, means morphological, these characters of the male and female is different. Okay, having this size is different. So nematode develop the larvae showing the four distinct stages of the growth during the life. Means during the nematode life, the larvae develop into four stage. So first larvae develop the inside the eggs laid by the female nematodes. Okay, then it undergoes molting inside the eggs, and second stage larva emerge from the eggs. Means these larvae directly infect to the egg of the nematodes. So these larvae emerge out from these infected eggs. So second stage larva enters inside the host, means host crop, that is a wheat, wherein it's developed, and third stage and fourth stage larva results in successfully. Means these are the four stage larvae which differentiate into the male and female nematodes. Means female nematodes lay eggs after the copulating with males, and interestingly both the adults soon dies. Means after male and female copulation takes place, so these adult means maybe female adult and male adult, so both the adults are dies, only eggs having the uh, nematodes are present, and these larvae directly hatch from these eggs into second stage larvae which remain inside the cockles or galls. Means these larvae within present these infected seeds or galls. So these larvae carries on the life cycles. Okay, so this is the uh, structure of the nematodes. So in this case, infected seeds having the larva can develop the initial infection on the wheat seedlings and mature plant. Okay, so in this way, primary and secondary infection takes place. Means at the time of the grain mature or milky stage of the uh, grain. So these larvae directly feed these seeds, as well as the milky content within these seeds, or directly infect to the leaf and other parts of the wheat seeds. Means this larva can remain alive and effective up to 30 to 40 years within the seeds or maybe on the soils. Okay, then last one, that is control measures, that is a cleanness. Seed is used for sowing purpose, or healthy one seed is collect from the disease free wheat plot. Then suppose this means before the use of the seeds for planting purpose, so these seeds collect from the infected plants. So these healthy one seeds and this infected seeds means galls. So these seeds may be mixed together. So easily differentiate or separate these infected seeds and other seeds by using the sieve methods. Mean just take the seeds and sieve the seeds. So different mesh size, so easily separate the infected seeds and healthy one seeds. So healthy one seeds is collect from the sieves and used for planting purpose. Then these seeds is soaked in the brine solution, that is a 20% common salt solution, by dissolving the 40 IBS of the salt in 25 gallons of the water. Means just take the 2 kg common salt dissolved in 10 liters of water, and these wheat seeds generally soaked in this 10 liters water having the 2 kg common salt. So after 5 minutes or 10 minutes, these infected galls or infected seeds is just float on the surface of the water or the solutions. So remove these infected galls or seeds and just take the seeds generally kept at the bottom side of the containers. So these seeds is removed from this pan, and these seeds is treated with the any nematicides or hot water treatments. Already I discussed the hot water treatment. Then crop rotation with non-host crop, because this nematode is survival in soil or seed is never near about 30 to 40 years. So that's why this crop rotate with other legume crop or cash crops. Then nematicides such as DD mixtures or nemagol or hexanema or nemaphos. So these nematicide just applied at the rate of 5 to 10 gallon per acres have been tested to control the nematodes disease seen in the wheat crop. And growing resistant varieties Sonora 63, Mexican blood, NP 908 and NP 227. So these some variety is somewhat resist to this nematode disease seen in the wheat crops. Okay, so this is the 4 diseases seen in the wheat crop. Okay, so first one loose mud, second one kernel bunt, third one leaf blight, and last one air cockle disease. Okay, air cockle disease means these infected airheads looks like as just a cockle, so that's why this disease is known as the air cockle disease. Okay, so any doubt in today's lecture, so ask me personally or send the SMS my personal mobile number. Okay, now I stop the my today's lecture.